Hello, everybody, and welcome to this pre-recorded presentation of Tranquis Therapeutics for the Biotech Showcase being held in January of 2022. My name is Sanjay Kekar, and it is a pleasure to be with you here today. I'll begin with a short overview of the company and briefly describe our strategy before moving on to describe our scientific approach, as well as sharing some of the in vitro and in vivo data in support of our platform targeting immunometabolic pathways. I'll conclude with an overview of our pipeline and describe some near-term milestones. So Tranquis Therapeutics is discovering and developing innovative medicines with the potential to revolutionize the management of neurodegenerative and aging-related diseases, and in so doing, dramatically reduce the burden these illnesses place on patients, families, and societies worldwide. Our novel therapeutic approach targets a key regulator of metabolism and mitochondrial biogenesis that we have shown is downregulated in myeloid immune cells as a fundamental pathological process underlying a range of neurodegenerative and inflammatory diseases. And this forms the basis of the Tranquis platform, from which we have discovered and are developing a pipeline of novel small molecules, which is led by our clinical stage compound TQS-168, currently in clinical development and positioned for ALS, as well as other orphan neurodegenerative diseases. Our fundamental innovation stems from the groundbreaking research from the laboratory of Professor Ed Engelman at Stanford University, which demonstrates that altered metabolism of myeloid immune cells underlies a variety of neurodegenerative diseases, such as ALS, frontotemporal dementia, Parkinson's disease, Alzheimer's disease, amongst other neuropathologies. We completed a $30 million Series A financing in July of 2020 with participation from SR1, Remages, and Vivo Capital. And this enables us to complete phase 1 and clinical biomarker studies with TQS-168, bring forward the next generation of compounds in preclinical development, as well as further develop the underlying platform. We have a strong IP estate with granted and pending composition of matter, use, and formulation patents. In addition to an experienced board, we benefit from a highly capable team covering the key disciplines needed for effective therapeutics R&D. In the interest of time, I'll not be able to describe our team in more depth. So as mentioned, we are an immunoneurology company targeting PGC1 alpha to restore dysfunctional myeloid cells. PGC1 alpha is an important master regulator of cell energy metabolism, and it's known that it is downregulated in patients with neurodegenerative diseases. Now, we know that myeloid immune cells, microglia in the brain, and macrophages in the periphery are important for CNS normal physiology and homeostasis. It's known that dysfunctional inflamed myeloid cells are a powerful, early, and continuous common pathway in multiple neurodegenerative diseases. So our innovation stems from the fact that the Tranquis scientific founders demonstrated that PGC1 alpha is downregulated in myeloid cells in neurodegenerative disease models, and that upregulation of PGC1 alpha can restore normal myeloid cell function, reverse inflammation, and have the potential to be a major breakthrough in the treatment of neurodegenerative diseases. From that, we've developed a platform for discovering orally bioavailable small molecules that upregulate PGC1 alpha in myeloid cells with potential application to both neurodegenerative diseases and non-CNS indications. That platform has yielded a pipeline of compounds and clinical and preclinical discovery stage candidates at various stages of development, initially targeting orphan and prevalent neurodegenerative diseases. This intersection of dysfunctional metabolic pathways, immune cell phenotypes, and a major unmet clinical need represents our core research strategy. So, as some of you may be aware, neuroinflammation is observed in multiple neurodegenerative diseases. For many years, conventional wisdom suggested that this observation was an epiphenomenon and not core to the underlying disease process. However, this understanding has been challenged recently, and it is now understood that myeloid immune cell dysfunction plays a critical and causative role in these disease processes. We know that neurodegenerative disease begins with a specific injury or defect that results in deposition or accumulation of toxic material, such as iron, amyloid, or cholesterol, which in turn induces an inflammatory myeloid cell response. Myeloid cells normally perform a cleanup function in the brain, taking up and breaking down everything from dying or dead cells to foreign matter. However, if overwhelmed by these toxic stimuli, they become inflamed, secrete a range of tissue-damaging cytokines, thereby initiating and propagating a cycle of neurodegeneration. This has now been recognized as an early and powerful pathological pathway common to multiple neurodegenerative diseases. So our unique approach identifies myeloid immune cell dysfunction within and outside the CNS to be at the core of the disease process. We identify and target master metabolic regulatory pathways that give us the ability to resolve the inflammatory state of these cells and significantly enhance their physiological function in the brain, for example, in microglia, and in the periphery, for example, in macrophages. Our approach has yielded drug candidates designed to not only stop myeloid cells from releasing inflammatory mediators, but reprogram them to shift back from a pathological glycolytic state towards the healthy oxidative metabolic state, and so by performing their normal physiological function, taking up and metabolizing the offending toxic stimuli to which I referred earlier. Taking an approach focused on this immune cell phenotype, we have designed, synthesized, and screened a series of compounds that comprise the Tranquis portfolio. Our lead compound is TQS168, which we've shown is orally bioavailable, crosses the blood-brain barrier, upregulates PGC1 alpha, and restores normal myeloid cell function with compelling effects in multiple disease models of ALS, frontotemporal dementia, Parkinson's, and age-related neuropathologies such as Alzheimer's. Furthermore, we've identified an on-target mechanism-specific clinical biomarker of the effect of TQS168, which will accelerate its clinical development and reduce risk. So moving on to some of the in vivo data, many of you will be aware of the SOD1 model, which is a standard but sometimes challenging preclinical experiment for the detection of a potential therapeutic signal for ALS development candidates. We dosed TQS168 in multiple repeat models of ALS at both Stanford and at Tranquis, where we demonstrate a highly significant improvement in both function and survival. We clearly demonstrate a statistically significant improvement in a key outcome in the SOD1 model, namely a significant extension in survival from birth. Other examples of the effect of TQS168 in the in vivo setting include the TDP-43 model of ALS and frontotemporal dementia, where we've tested the compound initiating treatment after disease onset. As for the SOD1 model, TQS168 demonstrates a dramatic improvement in survival, more than doubling the average lifespan in this model. We believe that this is a very important response in this highly challenging model of ALS and frontotemporal dementia. We also show a similar effect in the transgenic alpha-synuclein model of Parkinson's disease. So, with regards to some of the in vitro data, well, in BV2 cell microglia, the target cell type, we demonstrate a clear relationship between exposure to TQS168 and upregulation of the target gene expressing PGC1 alpha, shown up here on the top left. This is both a concentration and time-dependent response. The upregulation of the target gene corresponds with the reversal of the inflammatory phenotype in myeloid immune cells, again shown here in the top right in BV2 cells, where we show a concentration-dependent effect inhibiting TNF-alpha secretion at nanomolar concentrations. We observe the same responses demonstrating the ability of TQS168 to suppress IL-6 secretion, again at nanomolar potency. So the proof of principle we've been able to establish preclinically is a very important feature of the Tranquis program. In essence, we've been able to demonstrate that TQS168 is able to upregulate the gene expressing PGC1 alpha, a master regulator of cell energy metabolism in the target tissue, namely myeloid immune cells. In so doing, 168 demonstrates a significant improvement in survival and function in neurodegenerative disease models, such as the SOD1 ALS model, along with a corresponding reduction in the presence of inflamed myeloid cells in the CNS of subjects in these studies. Finally, we've shown that inflamed myeloid cells are increased in both frequency and activity in the blood of ALS patients. This biomarker taken from blood samples from patients is correspondingly resolved in a dose-dependent manner when incubated ex vivo with TQS168. So with our technology, we are able to demonstrate target engagement with a biomarker from patients, which is specific to TQS168 mechanism of action, establishing target engagement. And that target engagement corresponds with improved survival and function in established animal models of disease. So our overall R&D strategy is focused on two key drivers of growth and value. We've recently initiated phase 1 with our lead compound TQS168. This compound is positioned for orphan neurodegenerative diseases, ALS being the first target indication, but we're evaluating various other diseases where this mechanism has relevance that will act as fast follow-ons from that lead indication. In addition to the opportunity presented by 168 as a standalone asset in these indications, it also serves to prove in early clinical development the overall concept of targeting PGC1 alpha in myeloid cells through the associated use of a mechanism-specific peripheral biomarker. In addition to TQS168, we have a follow-on compound in development, namely TQS621, which targets the same mechanism, but is positioned for broader indications such as Parkinson's and Alzheimer's. And the execution of that strategy over the next 2 years will result in us moving from the early clinical stage today to being in mid and even late stage clinical development of 168, as well as in early clinical studies with TQS621, and with potentially 2 to 4 next generation compounds at various stages of development from lead optimization through phase 1. So during the next 18 to 24 months, we anticipate hitting multiple inflection points, beginning with the completion of the currently ongoing healthy volunteer and biomarker studies with TQS621, and move into ALS patients during the second half of next year, as well as completing preclinical development of 621 and nominating 2 to 4 next generation leads. So in summaries, ladies and gentlemen, I hope that we've been able to present today and show that we at Tranquis have leadership in targeting myeloid immune cell dysfunction focused on immunometabolic pathways. And our leadership today is represented by our discovery and the work, the platform we've taken forward around the understanding of PGC1 alpha biology, which, as we know, is a master regulator of cell energy metabolism, but we have demonstrated has broad applications in neurodegenerative diseases by virtue of its dysfunction in myeloid immune cells. This platform and this approach has led to a pipeline of already bioavailable small molecules led by TQS168, which is in clinical development, and then rapidly followed up by TQS621 for broader indications, as well as a portfolio of compounds which will lead candidate lead compounds through the course of the next year. We are at a clinical stage of development, as say initially targeting orphan indications, but rapidly following the broader indications. And we have a broad opportunity with multiple inflection points being generated over the coming years, and a substantial team which is growing, backed by significant investors who are supporting us in our mission. I thank you very much for your attention, and I'm happy for any follow-ups that the conference organizers may be able to facilitate. Thank you very much. Goodbye.